Hi students, Assalamu alaikum. Today our topic is on meiosis. First we define cell cycle. Cell cycle undergo the sequence of the change. In this change, first cell grow in size, secondly DNA of the cell become double, and the third cell divide themselves. And cell cycle consists on interphase and M phase, two main phase, interphase and M phase. Interphase consists on G1 phase, S phase, and G2 phase. M phase consists meiosis and mitosis. 30 hours required for complete cell division. From 30 hours, 23.5 hours required in interphase and 30 minutes required in meiosis and mitosis. In interphase, G1 phase cell grow in size of the cell become bigger, and in S phase DNA one from the mother and one from the father become double. Duplications of the DNA take place, and at the third G2 phase cell prepare themselves for complete division. Cell go into the M phase. In M phase, meiosis 1 and meiosis 2 for complete cell division take place in meiosis 1 and meiosis 2. In meiosis 1 consists on prophase 1, metaphase 1, anaphase 1, and telophase 1. In these phases, cell one by one divide themselves. And meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2 is like that of the mitosis. We discuss detail. Meiosis 1 in prophase 1 is a prolonged phase, consists on lipotine, zygotine, pachytine, diplotin, and diagnosis. First we discuss meiosis 1 prophase 1 of the lipotine. Lipotine DNA chromatin, DNA material, DNA chromatin change themselves in thin prolonged thread-like structure. This thin prolonged thread-like structure named this chromoneme, and fine bead-like structure appear on this chromoneme. We name this chromomere, and this chromomere in zygotine, the chromomere which have same genetic material, the gene location is present, the gene present on the same location, color gene, hair gene, skin gene is present on the same location, they come close to each other. We call this bivalent. They form bivalent. The color gene, hair gene, skin gene which present on the same location as compared to the other same location, both these location of the gene, eye color, skin color, hair color, etc is same, they come close to each other, we name this bivalent. And they we call this sister chromatide. Another name is sister chromatide and homologous chromosomes. And this chromatid as compared to the father chromatid as compared to the mother chromatid is non-sister chromatid, because the locations of the gene is not same. Location of the gene is not same as compared to each other, we name this non-sister chromatid. In pachytine, the sister chromatide pair with each other with the help of the protein, kinetochore protein. They pair with each other at the point of the centromere. At the point of the centromere, kinetochore protein pair the sister chromatide with each other. These sister chromatide we call this homologous chromosomes. Now these sister chromatide become homologous chromosomes, and the pairing of the sister chromatide is called synapsis. This process is called synapsis. And now this chromosomes name is homologous chromosomes, and this also homologous chromosomes as compared to these both to each other is non-homologous chromosomes. But this at singly we see, if we see at single, this is homologous and this homologous. If we compare with each other, these both non-homologous to each other, and they form a complex structure. We name this tetrad. Tetrad formed in Pachytene phase. In diplotin phase, student, these homologous chromosomes cross over with each other. The gene of the father overlap with gene of the mother, and this overlapping or crossing over take place, and segment of the gene exchange. This exchange of the gene cause the change in the children. Children is not totally same as the parent, because due to this crossing over, and brother and sister not physically, mentally same due to this gene exchange. In phase of the diplotin, gene exchange take place, segment of the gene change take place, father gene overlap with the mother gene. This point this point is named as chiasmata. This structure is called chiasmata formation take place, and the crossing over cause the recombinations of the gene. This process is called crossing over. In diagnosis, centrioles disappear and cell membrane start disappear, and nucleoli become disappear, because no cell prepare themselves for complete division. That's why the centriole cell membrane become disappear. Now the translocation, the separations of the homologous chromosomes is called translocation. Now translocations of the gene take place in diagnosis. Some father gene translocate to the mother chromosomes, and some mother chromosomes gene translocate to the father chromosomes gene. So this is called genetic recombination, and this is called variety change. In metaphase 1, prophase, after prophase 1, metaphase 1 start. In metaphase 1, the kinetochore protein which attach on the centromere of the chromosomes is attached with the spindle fiber of the centrioles. Centrioles spindle fiber are kinetochore fibers attached with the kinetochore protein of the centromere on the centromere of the chromosomes, and place these homologous chromosomes in the equatorial. Arrange the bivalent chromosomes at the equatorial plate in metaphase. In anaphase, these chromosomes, this kinetochore protein and kinetochore fiber or spindle fiber start become short and separate these bivalent chromosomes or homologous chromosomes from each other due to the shortening or due to the compressing or stretching of the spindle fiber of the centrioles. Kinetochore fibers which pull individual chromosomes to the respective pole. So kinetochore fiber pull these individual chromosomes to the respective pole. In telophase, the cytoplasm become divide. This process is called cytokinesis. And nuclear membrane reappear, centrioles become reappear, nuclei become reappear in telophase. In telophase, cytoplasm, the membrane of the outer membrane of the cell invaginate and cytoplasm divide. So this not complete division take place here, because the diploid chromosomes present here, not haploid chromosomes. In meiosis, in definition of the meiosis, the number of the chromosomes in parent cells, number of the chromosomes in daughter cell is half as compared into the parent cells. So this chromosomes number is diploid, not is in haploid. In like this father, this is diploid in nature, diploid, and this is diploid. So this cell need more division for become four haploid cells. So in meiosis 2, more division take place here. In prophase 2, the in prophase 2, the centrioles again disappear and the nuclear membrane become disappear, because cell prepare themselves for division. And in metaphase 2, again the spindle fibers attach with the kinetochore protein of the individual chromosomes and place these chromosomes at the equatorial, arrange these chromosomes at the equatorial range. And in anaphase 2, the spindle fibers of the centrioles stretch themselves to the respective pole, stretch and um, and individual chromosomes lead to the respective pole. And in telophase 2, the complete division of the cytoplasm take place. Cell divide themselves into two. One cell divide themselves into two and other one cell divide into two. So this process is called cytokinesis. Division of the cell is called cytokinesis. And this 23 chromosomes, 23 and 23, 4 haploid cells is formed at the end of the telophase 2. Now this cell is haploid, and meiosis phase is a reduction phase. In this, number of the chromosomes is become half as compared to the parents. But in mitosis, number of the chromosomes in daughter cell is same as compared to the parents. So in parents is a 46 chromosomes present, 23 from mother and 23 from father, 46. But at the end of the meiosis, I have reached 23 chromosomes. So this is haploid chromosomes, and 4 haploid cell is formed. And when this cell become mature, they become chromosomes, 33 chromosomes, and 23 chromosomes when during fertilization attached with the 23 chromosomes of the egg, and they become female child. If the chromosomes is X and egg is also X, after fertilization female child is produced. And if this Y chromosomes, haploid Y chromosomes fertilize with the haploid egg X. Then male child is develop. So this is meiosis. Hope you understand this. Like and share. Love is.